information is much more complicated and still, I don't want to say design, but there's order to the chaos. And that means that you can create a lot more variability. So for example, you have DNA, it gets transcribed into RNA and gets exported into the nucleus. Once you have that piece of RNA, it gets bound by a bunch of stuff and usually gets translated if it's an mRNA, but sometimes it will get arrested and then brought back into the nucleus. At that point is when these reverse transcriptases will flip it from RNA to DNA and start opening up many areas of the genome. When you open up the genome, you transcribe it. So usually in eukaryotic systems, the entire DNA is completely coated with histones and is very closed. But the areas that are specifically being used in that specific cell, because keep in mind, every cell type is opening up different areas of the genome and using different areas of the genome, can then react to its own state, cellular state. So it opens up a lot of possibilities. It really makes it far more complicated and lets you do many, many more complicated things. And that's why one of the things that was very weird when people sequenced the human genome, they expected to have like 200,000 genes. They expected far many more genes than they actually found. And in reality, there's about 20,000 protein coding genes. And they still can't really explain where this like extra complexity comes from, especially when you compare it to things like bacteria.
Time to talk with me. I appreciate it.
Gladly, of course.
When I got your email, it was interesting because like you had mentioned about epigenetics and coming across your work spontaneously that way, it got me back into the whole phenomena of epigenetics in general, because it was a difficult thing to navigate looking into, from especially not being an academic. It's like, you know, a space that's hard to get into as like an amateur researching these topics, and then looking into your work on it. It's such a fascinating endeavor.
And honestly, I wouldn't feel too bad. At least the vast majority of the biological field doesn't even pay attention to it. Basically focuses on the 1% of the genome that's coding, that is genes, and the rest is kind of like a nice thing to think about.
Junk, yeah, it's like they call it junk DNA in my high school biology class, and it was just codons and sections of code that were almost mistakenly added in by evolution, is what it was always, and then it was just kind of disregarded, it seemed, a little bit.
Is honestly from bizarre to me. The reason is I think it's kind of historical in that our understanding really comes from bacteria, and it kind of grew out of there. And bacteria are fairly simple, as in vast majority of their genome is coding. We're talking about, like, I want to give it 70%, more than that. But then when you get into eukaryotic systems, the story completely changes. And we're talking really like a vast minority of the genome are genes. So, which is weird.
Yeah, just the coming about of the nucleus in evolution, I guess, that right there caused this giant influx of genes that aren't necessarily coding for. And so you mentioned just briefly in your email at one point about the coding for viruses and stuff like that. If I guess what I would like to kind of ask a little bit about is, what are these genes doing in your mind, and what does epigenetics play? And epigenetics as a term seems kind of anytime anybody puts the prefix epi on anything, it's like a way to not a way to circumvent by any means. In fact, it's a way to come at it and looking at it. But it's like in philosophy, they always use the term epiphenomenon to kind of get around consciousness, to kind of like sidestep the whole issue of mind. But epigenetics seems like a way to actually do the opposite in biology. It's like to actually step into a field that's completely emerging. What do you see what does epigenetics mean really to you as far as a study?
Yeah, so I mean, the definition is anything that's on DNA that isn't a mutation would be the definition. The way that I think about it is, so there's this concept of central dogma that is literally the central dogma of biology, meaning, and what that is the kind of the flow of information in a biological system, which goes from DNA to RNA to protein, which is well understood, and is from DNA to RNA is transcription, from RNA to protein is translation. And bacteria do that, and that's they're almost like chemical little things. In eukaryotic systems, the flow of information is much more complicated, and you can't really think of it as a linear process, meaning it's not DNA to RNA to protein. It's really DNA, RNA, and protein form like a triangle, and they really talk to each other. So, for example, DNA will talk to itself by chromosomal looping, for example. So that means an area of DNA can impact another area of DNA, not through the central dogma, right?
I see.
So that is one piece of
Wow.
epigenetics. And that's kind of where you can modify parts of DNA in order to change how it interacts with itself. And then there's also RNA, which also can interact with itself. And then there's also proteins that also can interact with itself. And in reality, the central dogma posits that it's a linear flow of information. It's a one-way flow
Yeah.
of information. In reality, it's bidirectional. So when you have, for example, DNA and RNA, usually it's one way, meaning transcription. When you take DNA, you transcribe it to RNA. But there's also reverse transcription, and this gets at the viruses. We generally don't think of reverse transcription as being important for eukaryotic systems, but in reality, when you go look at the genome, there are reverse transcriptases encoded in viruses all over the place. And what that lets you do is starting to play around with things like you can open up specific areas of the genome by reverse transcribing the RNA and then importing it into the nucleus.
Oh,
So that
wow.
causes a lot of interplay and transcriptional programs that are basically making the cell go.
Yeah. Okay. Yeah. And that process being bidirectional. It's certain, like you said, the central dogma has always been, at least, in my limited biology education, has always been that the ribosomes are integral, and there's all these enzymes that are dissecting this basically template, or genomic template, that's kind of creating a phenotype. You got the genotype, you go to phenotype, it's one way, and it's this, like you had said, this transcription and translation process, and it's very machine like. It doesn't, it doesn't have these, because it seems to me like, if you reverse it, or if you go against that grain of the central dogma, you then open it up to a little bit of unscientific type phenomena, which is the whole point of investigating it, is to figure out what that really is. But it seems like then your chance of mutation and variation in the genome maybe become less than random, or there may be some other influencing factors potentially, you would think.
Right. So the flow of information is much more complicated, and still, I don't want to say design, but there's order to the chaos. And that means that you can create a lot more variability. So for example, you have DNA, it gets transcribed into RNA and gets exported into the nucleus. Once you have that piece of RNA, it gets bound by a bunch of stuff and usually gets translated if it's an mRNA, but sometimes it will get arrested and then brought back into the nucleus. At that point is when these reverse transcriptases will flip it from RNA to DNA and start opening up many areas of the genome. When you open up the genome, you transcribe it. So usually in eukaryotic systems, the entire DNA is completely coated with histones and is very closed. But the areas that are specifically being used in that specific cell, because keep in mind, every cell type is opening up different areas of the genome and using different areas of the genome, can then react to its own state, cellular state. So it opens up a lot of possibilities. It really makes it far more complicated and lets you do many, many more complicated things. And that's why one of the things that was very weird when people sequenced the human genome, they expected to have like 200,000 genes. They expected far many more genes than they actually found. And in reality, there's about 20,000 protein coding genes.
Jeez.
And they still can't really explain where this like extra complexity comes from, especially when you compare it to things like bacteria. And this is where the actual complexity comes from, the interplay of information between those three components.
Yeah, it's kind of wild too, because like you just mentioned, you've got all of these different specialized differentiated cells after embryonic development. Once stem cells have which is another really odd area, which I think may merge with your studies, is the idea of non-neural biological electrical networks that Michael Levin, for example, has been kind of looking into. I don't know if you've heard too much of his work, but what he's been doing in his studies is looking at what actually tells an arm to stop growing when it's reached its final length, or what cuts off the phenotypical arrangement of a genotype that, say, a stem cell's differentiating and it's becoming an arm cell, or it's becoming a finger or a muscle. Because it always is interesting to think, yeah, we have this giant code, like you just mentioned, of this infinitely long string of characters that's being transcribed and translated in a unidirectional central dogmatic manner. But when that is shown to be not the case, it's still interesting to know, like, what causes to shape the morphogenesis aspect of it. And so what his studies was looking at is that there's something about these non-neural biological electrical networks that exist in embryonic development and throughout, that, like, when a lizard's tail gets cut off, it grows it back. If we lose our arm, we don't. There's this, like, there's these gradients, is how he describes it. There's voltage differentiate, like these voltage gradients that exist, similar to when you just have regular osmotic diffusion, where you've got, more of salt on one side of the membrane and less on the other. So you've got this gradient of, so I don't think he's meaning like voltage necessarily like an electrical push, but more of just a gradient differential of ions. And his team began manipulating these gradients, and what happened was he was able to, long story short, because I'm not a biologist, is he was able to grow eyes on the back of a frog, basically by just not altering the genome or anything, but by altering the configuration of these electrical networks that basically determine the overall morphogenetic shape. And so it's kind of like, yeah, you've got DNA and you've got the transcription translation process, but ultimately there's still something missing from the equation. Like, like you said, like, what is these DNA almost has to reference itself, you think, because if it's not, what's it referencing when it builds its organism almost? Because it doesn't have the shape, that's kind of an odd thing.
And so it's actually one of the things that really stuck out to me. There was this paper that I read, and this 10 years back at this point. They looked at the pre-implantation embryo, so specifically the blastocoel. So it's, as you get the egg, the sperm emerges, zygote, you get 2-cell stage, 4-cell stage, 8-cell stage, and onwards. You go look at each cell stage, and this is when you start seeing, like, invagination, you start seeing the development of the embryo. At each stage, there's a specific set of viruses that gets turned on. And it's very specific. So these sets of viruses are the ones that control which sets of genes are getting turned on or turned off as it goes forward. And I think that this is kind of, my intuition is pushing me in this direction, that the sets of viruses that are turned on are electrically sensitive. So that means that the electrical profile of that cell will actually change what viruses are getting turned on or turned off.
Wow. And then those viruses, once they're turned on, their function is to assist with, when they get turned on, is to assist with this process of
orchestrating the transcriptional program of the cell.
Wow.
So the viruses themselves, as they get activated, right? So when the 2-cell stage, I think it's at the 2-cell stage, the entire genome gets wiped. So you have about 2 meters of DNA inside of each cell nucleus, and that 2 meters of DNA is the same for every cell, but it's folded differently for every cell type, and it's folded differently perfectly for every cell type. That folding is controlled by these viruses, and as it goes on, it changes and defines its own program, if you will.
Yeah, okay.
It feeds back on itself. And as you can imagine, when you're doing reverse transcription, you're creating more DNA that then goes back and does stuff. And the DNA that gets reverse transcribed then gets transcribed again. And as you can imagine, that causes a system that is extremely complex from relatively few parts, which is, in my head, very beautiful.
Yeah, it's like a combinatorial explosion at some point. It's like you've just got this massive quantity of, and that's how you would get complexification from such a small amount of nucleotides, is you would have something as dynamic and as intelligent as that process seems to be. And it's like in biology, you're generally kind of shown, at least at the elementary level of biology, that things are orchestrated by enzymes that are built, and you don't hear about these viruses and about, because it's critical, it seems to me, that if there's viruses that are doing this, that without knowing that, and you saying it's has an electrical field, I think that the work that I was mentioning earlier, that I kind of butchered its description, but what it is and its inner workings is looking into how these biological and we usually focus on electrical impulses with neurons and specialized brain cells or central nervous system cells. At a minimum, we do that, but then we seem to forget about the electrical signals in somatic cells and cells that are body only. And so I think your intuition there about how electrical fields in the early developing embryo and everything do indeed influence the actual phenotypical expression of genes could be something very unique about that.
And when you get into the brain, it is freaking fascinating, because if you pick one neuron and you pick another neuron, the chances is that they're genetically unique to each other, which is something that again goes against the central dogma. The idea is that every cell within your body has the exact same genetic content. That idea is completely wrong, especially when you look at neural cells. And it's wild because you go look at, for example, let's take a receptor like GABA, for example. Yeah. You go look at GABA receptor and you can see that and I'm talking about go look at the genetic loci, which is, most of it is non-coding. You go look at the genetic loci, and within the introns of the gene, there are these viruses that again are reverse transcribing. So what you actually see in neurons is that when these areas of the gene gets activated, it can actually change the copy number of that specific receptor. So as you use that neuron, it will change it's own genetic components. And I would say that's a rudimentary form of memory, essentially.
That's what it sounded like almost, because, memory is such an interesting thing, but of course, it's also crazy because, with dreams and all of that, it just seemed like there's a little bit more to dreams than random neuronal firings. There's a little bit more to memory than simply recalling through this layered, hierarchical structure up to the cortex, which there may be, but there's billions and billions of neurons. And so to think each one of them may be dynamically and genetically being altered per memory, it could really, really put some credence to memory as a whole new phenomenon. Than just in psychology, you look at it as this encoding to long term. You've got flashbulb memories where you remember episodically like emotionally stimulating things because the amygdala is triggered at the same time as the hippocampus or something like that. But there's more to memory, it seems like, than these 2 or 3 types. It seems like a dynamic thing, and the epigenetics of the brain is highly wild.
So that means that as you memorize things, as you go through life, you're changing your genetic components, and that means that neuron is going to react differently, right? If that neuron says, okay, I'm going to take GABA and make 10 GABA genes, it's going to be producing more receptors on the outside of it, so it's going to change its function. So as you go through life, you are memorizing things in DNA, which changes the neurons, which changes the electrical components. And then this gets at, so you talk with Kelvin, and one of the things that was very useful in his theory is that there's a clear distinction between mass and matter. DNA is matter. Brain waves are mass. So you're able to transfer mass and matter and actually make matter in a way inside of your own brain, which is wild.
And talking with Kelvin, it's been a while, and even when I spoke to him, it wasn't. It was still a difficult theory for me to put my head around. And he does a great job, and he's really willing to help, chat and figure it out if you have questions. And so I got some headway on there, and then I haven't returned to tetraionics as an idea in a while. So I was gonna ask you about that. What doors did that kind of open? I know you just said the mass and matter. What do you remember specifically kind of how he configured that idea? I mean, I can't recall exact. I know it intrinsically to me, it seems like energy and mass are equivalent with the speed of light as a constant, E equals mc squared, the equation. And there always seems intrinsically like energy and mass are separate phenomena, but mass and matter, he differentiates that with the equilateral triangle. Do you mind describing that again,
Yeah,
or if you
it's
don't mind?
actually almost like depressingly simple, where it's
Yeah,
mass is 2D, it's waves, it's fields of energy, right? Matter is 3D, it's particles. That's it. That's literally it. One is
yeah, okay,
2D and one is 3D. What it let me do was really, like, when I first stumbled on his theory, it was, I guess, now like 8 years ago, and it seemed like a curiosity that I couldn't put down.
yeah,
Honestly, I've gotten myself into trouble a bunch of times because when I get interested in something, I will not put it down until my curiosity is satisfied.
yeah,
So what I did was, what it offers is, unlike any other unified theory of physics, it actually lets you build up. It lets you go from 2D fields of energy, which are the triangles, right? 2D little triangles, which is energy. Each quanta is made up of,
quanta.
right? It's a quanta of
Yeah,
energy.
like a quantum coin is what he was showing me in the
Exactly.
actual tactile models. Yeah,
Right. And then from using natural order of things in terms of like negative pairs up with positive, and then you start building up from there, you can start building tetraons, which are little tetrahedrons. And then from there, you build up into quarks. And then from there, you build up into protons and neutrons, aka baryons, as well as leptons, electron. Then you build into deuterium, then you build into elements, then you build into small molecules, such as nucleotides, and then you can build it into DNA. Once I built it into DNA, it was clear that this theory was able to produce a structure that has been experimentally verified using, X-ray crystallography. So
wow,
that at that point, it allowed me to think about these things not as like kind of ethereal balls that are coming into contact. It's crunchy. This
yeah.
is the way that it looks, and that causes a lot of other things that you can start thinking about. For example, the mass versus matter component. So
Yeah, yeah, exactly. Yeah, you start looking at, yeah, it changes the dynamics of a lot of this. How has I guess discovering Kelvin's tetronics and then putting it into this model, the video game that you created that I was looking into briefly. I haven't which I'm going to try to get into more. It's fascinating. It truly is. Have you used some of the dynamics and physics, I guess, from Kelvin's ideology to and kind of create that? I mean, I guess you created that game, right? Which basically is what is that game exactly, if you don't mind?
yeah, so in like, I've been told so many times by multiple people now, stop calling it a game. It's really a model.
Oh yeah, yeah. I wanted to
It's
call it a game just to give
right.
you the credit that because that's
It
actually
should
hard
be
to
a
make
game.
a game. So I mean, it's not like it degrades
Right. I
the
mean, I'm of the belief that the best way to learn is to play. And
yeah,
then this is the point. What it is basically a model of the eukaryotic cell. Specifically, I would say the metazoan cell.
okay,
And that lets you think about these processes, these chromosomal looping. So that's that's at MIT, I studied chromosomal looping under Richard Young. And that, as you can imagine, is a physical problem, right? It's a problem of having a piece of DNA with a bunch of proteins bound that then chromosomally loops and then influences itself. When it's a physical game, right, you need to be able to look at it. You need to be able to come to some type of conclusion as a human in order to look at these structures and figure out what's happening. And that's the point of the game. In terms of taking a cell that is doing some process, it's differentiating into an arm or whatever,
yeah,
right? How is it doing that? How does the 3D shape of the genome actually cause that to happen? And that's
yeah.
what the point of the game is.
I see. Yeah, to get a visual kind of process to envision what this really is, because when you think about that looping aspect of it, it's kind of self-defeating. As anything really, when you get into anything that works, it's like you get to the crux of it and you find this loop of referential processes that seem to ultimately philosophically depend on something transcendental or something that there's this ultimate truth that things are beckoning towards whenever they produce a 3D shape or anything. And that's why those morphogenetic aspects of like, how does something assume its shape, and how does something not merely differentiate into different cell types, but how does it know to stop growing? I mean, yeah, we have start, stop codons and things like that within this structure, but that's too simplistic to describe some of these processes.
Right, and I would say that biology right now is kind of floundering. They cannot. So my background is in genome-wide association studies, which are GWAS for short. Basically, what that does is it sequences millions of people and then puts them into two buckets: with disease, without disease, and then finds which areas in the genome are correlated with that disease. Now, the expectation was that all the hits, all the areas that are correlated with a specific disease are coding. And unfortunately for the current field, almost none of them are coding. Like, we're talking about like sub 1% of the areas are coding.
So that you can't even do science on them at that point, or in the same way.
Yeah. Right? What do you do? How do you interpret this? How do you design a hypothesis backed by these correlations, right? Correlations, not causation. How does it actually cause these different phenotypes? So building from the bottom up in order to understand these looping things was key for me. And I've always been obsessed with 3D shapes and structure. And that's a lot of what Kelvin's theory let me do. And once I built DNA, once you actually look at DNA built from the bottom up, and it looks like DNA that was measured, I couldn't put it. I mean, at that point,
at that point, you, especially with your field of expertise already, coupled with that kind of rigor of evidence, you can't. You would be disheartened if you were to put that down, I would assume.
Right.
And those Platonic solids, like the tetrahedron, those, like, I mean, they're called Platonic solids for a reason. I mean, Plato was in the Aristotelian metaphysics back in the Greek classical days of philosophy being written. You've had ideas that hinged upon these types of shapes for some reason.
Right.
You find that interesting? I mean, that's kind of wild.
Right. And you, like, at some point or another, the number of coincidences becomes too great, and you can no longer ignore it. And I mean, I can get into this, but one of the things that also the theory let me do, one of the key things that it does is it says that each element has a specific number of protons and neutrons. The number of neutrons does not change. So that means, like, carbon has 6 protons and 6 neutrons. That's however many it has. That actually kind of let me figure out a little code that appears to have been embedded in an organism that has been around for a long, long time. And this gets into where the nucleus came from. The idea currently is that it kind of originated from magic, almost, in terms of it spawned from bacterial ancestors about 540 million years ago. In reality, it appears there was an organism that fell on Earth that had a code embedded within it that you need to use tetrahedronics, or at least understand how the physics works, in order to decode. And that helped a lot in my understanding of speciation and evolution, and these things I'm mentioning about viruses controlling what defines a species, etc.
Absolutely. Yeah, no, you just mentioned, speciation, micro to macro evolution, species to species. And, coming once again from my high school education, it's biology referred to speciation as only being happening by literal geographical separation, which never fit for me. That every time there was this split, there's thousands of millions of species. There wasn't earthquakes and like Grand Canyons being formed left and right that were just geographically separating species when they speciated. And even if you get to that point, it's still difficult to justify something with unity becoming multiplicity out of just that. Like, you're introduced,
right? It's weird.
And I'm not saying, I mean, I think evolution by natural selection, whatever nature is that can select. Once again, I think natural selection does imply there's a selector, which makes it more difficult to justify. But it's the best kind of grand unified theory of biological diversity that we seem to have, and it does form like this backbone of biology.
Exactly.
So
And
it
it
is,
works.
and it works. And so when you mentioned speciation there, what about this idea helped you kind of justify speciation or see speciation as a possibility? I know you probably just
Yeah.
explained it a little bit, but
So when you have the viruses that are defining the transcriptional program, right? And you think about a virus, you think about your immune system, right?
yeah,
So how do you define what viruses are your own versus everything else, right? That is the innate immune system. So that hooks the innate immune system into speciation, because if they're your viruses, they are not monkey viruses. They look slightly different and behave slightly different. And you've built an immune system to recognize your own viruses versus everything else. So that allows you to clearly define species as you move forward. And also, this gets into the Darwinian versus Lamarckian debate. When I talk about your neurons genetically changing over time, that is not Darwinian. That is Lamarckian. And that,
yeah, the use and disuse, is that what Lamarck is? Sorry to
right,
interrupt you.
exactly.
Okay, yeah,
Yeah,
like the giraffe's neck would get longer if it used it and stuff. Basically, the use and disuse. Okay,
use and disuse of not your neck, but use and disuse of specific viruses.
yeah,
And that trickles into speciation because it controls, like, you can change the methylation of your I'm looking for the word, but I mean sperm and eggs. You can actually change the methylation, which changes which viruses get activated when the organism is developing. So that means that what happens to you can actually change how your offspring is. And a good example of that is grandmothers who have gone through starvation periods will actually have granddaughters who have a higher propensity of being obese. And how exactly that information gets propagated has been a very hot debate for a long time. And now they're starting to figure out that the way it gets propagated is through methylation, which controls the expression of these viruses.
really. Okay, so yeah, the methylation. Okay, yeah, which way to? Wow, that's wild. And I'm sure there's probably innumerable examples of this being found. I guess where it comes down to is, as clearly as that's being demonstrated, people, it just seems like this creates a big divergence in biology, because once you do, which this space is so critical, and your work is just, I assume that it's in scientific academia, it's difficult.
Well, it was not greeted well. Let me put it that way. It got me in a lot of hot water, and
Oh,
as far as I'm concerned, I've traveled in the upper echelon of scientific circles in terms
yeah,
of. I was at Carnegie Mellon, as at MIT, at the Broad Institute of MIT and Harvard. The people in my thesis committee were really like, I mean, Eric Lander was on there. He was the chief science officer for Biden for a while. And it's the dogmatic view of the central dogma has become so ingrained in people that they're unable to see outside of it. And I understand in a way, because it's scary, in that, like, right now we have a very clear picture of this is how it works. And then the other stuff is just little cute curiosities.
yeah,
But in reality, the cute curiosities are particularly important. And to me, it harps back to when Mercury, the orbit of Mercury was used to kind of disprove Newtonian theory, and because it was
yeah.
a curiosity, and then eventually became more than a curiosity that could not be explained away. And that's kind of where we're entering with biology, where people want to explain away all this junk DNA as being like a weird evolutionary blip. But in reality, every single species, every single metazoan on Earth, metazoans being anything that is divided, that is symmetrical.
Bilaterally symmetrical, I assume,
Mhm.
most cases, yeah.
exactly, has these viruses, every single one. So it's like,
That's wild.
you can't, like, how can you say that they're junk, that they're magic, that they're just doing nothing? And that was really disheartening, because that's the view that was pushed at MIT at the highest levels of learning. And that, in my head, actually is a real big problem. It's a problem that is bigger than myself. It's causing a lot of lack of belief in science, I would say, because science is ignoring some key pieces in order to make a simple narrative.
Yeah. Yeah, no, that's crazy. And to hear you about the hot water it's gotten you into and stuff like that is unfortunate, because of course, but that is the nature of science, I guess. It's almost like everybody in the previous
Exactly.
generation has to, I heard this recently, that like everybody in the previous generation of science has to like die before it. Unfortunately, it's like it's almost like people have to die, and not in a bad way. I'm just saying people die anyways, but it's like, it almost seems like for new ideologies to surface. It's like even with the history of, with the philosophy of science, you look at, there's philosophers of science, and I've talked with a couple of them, and it's interesting to trace the philosophy of science as being that in science, you've seen this, like, even with plate tectonics, it took all of this to see seafloor spreading and the movement of the Earth's crust, for that to enter the sphere took like 60 years, even though there was heavy duty amounts of evidence. And so in the philosophy of science and the history of science, it's like, we're almost guaranteed that everything we hold dear and factual right now, if it follows the same trajectory that we're on. If you were to draw this and graph this on a Cartesian graph of science history, you would have to admit all of this stuff that we hold fact now will probably be proven false in 100 years if we follow the trajectory as graphed out so far. And so I wonder how this will work.
Right, and that, in my head, that's really exciting, right? And
Yeah.
there was actually a paper that you bring up. I forget, they coined a term for it. When big superstars die, that's when there's new entry into the field, and you can actually quantify that using citations in specific fields.
Oh yeah.
Using, like, you basically divide it up by fields, and then you look at the number of new citations that are in that field, and whenever a person at the upper level dies, that's when new citations come rolling in.
Yeah, that absolutely. And with your work, being able to work with people in the scientific fields and those institutions of extremely high, some of the best, it sounds like, in the world, in the world, really, on some of these things. Have you found yourself at a place where you can, and this is just a personal question, really, have you found yourself at a place now where you're able to operate on this stuff without that? Just because I would, I'm sure you have a lot of passion for it, and I hope that someone like yourself gets to have their endeavors, fulfilled.
Yeah, so I mean, yes and no. What I mean by that is, really zero institutional support. Really, I was basically excommunicated from the academic community because of this.
Yeah.
So I had to find new people. In terms of, now I've gone through a bunch of startups in order to kind of recruit, find people, starting to talk about like what to do with this knowledge. So when I mentioned the chromosomal looping, it lets you understand how transcriptional programs are actually initiated. Once you understand how these programs are initiated, you can start playing around with them. You can
I see.
start editing epigenetics and changing the expression of gene X or Y. So that's what I've done in terms of, I'm partnering up with a couple of people and making a new company in order to actually edit these genes and turn up or down specific genes that are tied to specific diseases. So
Oh, okay.
yes and no, as in like the kind of like traditional way to go about it is completely gone, but it's opened up my ability to understand these things and build up.
Absolutely. Well, that's awesome. In that way, I mean, as rocky of a ride as it would have been, yeah, I mean, the possibilities are endless. And I think something about your intuition with these electric fields is something too, because like I think you'd be very interested in us, especially with your ability to comprehend it better than someone like myself, would be really interested in those non-neural bioelectric fields that are at the crux of Michael Levin's work in Massachusetts, I believe. I can't remember what university he works at, but his work has been, and he even admits to not really understanding where these come from and where these originate from, but basically they even have like a prenatal face, like the outline of it will indicate like the phenotype look of a face, and if you were to edit those gradients that make the shape of a face, then the organism is born with an eye a little bit out of place, just as a per how they alter these gradients on these. So I think that work may be commingling with yours at a distance, without even knowing, but it's fascinating to think of it like that, that there's work being done, in these areas, and there, but it's like you said, I guess it's kind of under recognized or underseen, but work like you, like persisting with it through these startups and all of that, it's not the easiest route, but it's the route by which phenomenal change occurs.
Right. And I kind of came to the conclusion of like, if not me, then who? Like, who? Right. So, like, if I saw somebody, like, if I felt that the scientific community was like trending in the right direction, that they're going to find these things, that the profit motives are not really impacting what you do look and for, then I'd be like, okay, yeah, that's fine. You know, I'll just go farm, I'll chill out. But right now, I think humanity as a whole is going through some really terrible times, and it's going to get a lot worse. And unless we figure out these things about like biology, about energy, about these key fundamental things that need to be overturned, because as you said, there's all these old people. I mean, all these professors are extremely old. We're talking about 70 plus.
Yeah.
And this is also true for the oil executives. It's also true for all different sectors. Unless there's a revolution in this kind of thinking, I think we're kind of screwed as a human race, honestly.
Oh yeah, I don't know. I don't, and like I said, I mean, I don't know a whole lot, but what I've come to think and realize is that consciousness in the West and in the world as a phenomena itself has been taken for granted. The fact that we have a subjective element and frame of existence by which we experience the world. And so science is this way of removing that element and looking objectively at the world as if we, as an experiencer of the world, as a seer of what is seen, do not influence what is seen. And epigenetics comes down to that too, is the idea of influencing this static genomic template is obviously, heresy to the strict central dogmatist. But looking at consciousness, it's critical to put the element of what is observed, back, the observer influencing what is observed, and that we look as if in science we're not a part of this world when we observe it, and obviously observation has been proven in physics at the lowest level to collapse wave functions, maybe, that something about consciousness, when things are observed, they go from quantum to Newtonian. There's something about conscious observer in the picture of things does influence the world. And I think maybe this has something to do with it.
It's interesting. So the tetraionics, one of the things that's key about it is it's a deterministic theory, meaning
okay.
So that means that if you observe something, it's not going to collapse the wave function unless you add energy to the system. And that means that whenever you're measuring, like we are in an ocean of triangles floating around constantly, they're called electromagnetic waves. So whenever there's any type of contact between one electromagnetic wave and another electromagnetic wave, it'll be measured, right? The only way to measure something is to touch it, to poke it, to see it, right? And that means that you are perturbing the system. So what tetraionics lets you do is that when you define the difference between mass and matter, it lets you understand that the things that are happening, right, the shape of an electron, the shape of DNA does not care about you. It will always be the way it is. Even if you look at it real hard, even if you look at it a different way, it does not matter.
And that makes sense.
Care about you.
Yeah, and that's kind of what I ended mentioning, the wave function collapse. I've had a lot of issues with the idea, especially like Penrose's theory. Roger Penrose, he has a theory that he's worked with with an anesthesiologist at the University of Arizona, Stuart Hameroff, and they've come up with this idea of orchestrated objective reduction, which implies that the wave function is perpetually and constantly collapsing all around you, emitting particles of consciousness. Now, but here's the problem with the older school of thought is everything's particulate, everything's got to be fit into the uncertainty principle, everything's, and that's where I detracted away from the Penrosean hypothesis of constant and perpetual wave function collapse, and that there's particles essentially of consciousness being emitted, received, and there's this physical materialist framework by which we're exchanging information with what we perceive. And I don't think that that's necessarily the right way.
And in reality, it's kind of both. So as in, you have an electron, right? In terms of an electron, is the 3 tetrahedrons, 3 negative tetrahedrons arranged in the shape, which is another coincidence. Have you know the sign for radiation?
Yeah.
That is what an electron looks like, just from the side.
Yeah.
So that particle, right, has vast majority of its energy not within the particle, but around the particle. So the black body radiation that it's emitting is actually what's coming into contact and touching things and making decisions. You could call decisions when you overlap one field with another field, it will react and produce something different. That's called a quantum computation. And then when you talk about what a brain is, it's a very complex, beautiful quantum computation that is able to record things in matter. So as you can see, it's both, right? In a way, you could say that there's consciousness everywhere and there's quantum computing happening everywhere. But in reality, it's a much more complicated system when you start talking about brain waves, about actually doing computation in a manner that is intentional.
Yeah, exactly. That's especially when you look at a deterministic system, like you mentioned, philosophy would require the discussion of agency, like individual will, free will. Do we have free will? Is that a part? But granted, does that really influence the scientific aspects of these advancements, like disease? What are your potential grand futuristic hopes that epigenetics and disease, especially when we look at mental health, especially mental health, that's such an enigma to begin with. When you start looking at the mind-body connection, consciousness, I mean, it already is not understood because psychiatrists are doctors of the mind, but nobody knows what the mind is or where it is, where it lives. Like you go to a doctor and you have a cut on your leg, you hope that he knows what the femur is, and you hope that he knows where the femoral artery is located, and he can show you that on an x-ray. But if you go to a psychiatrist, you're not like, hey, can you x-ray me and tell me where the mind is, And so seeing that, and then talking with a couple mental health professionals that are looking at, like Dr. Anup Kumar is a doctor I've had on, and he's a really good doc. He's a really cool, really neat way of integrating the mind and the body. And in fact, it's a triangular shape, once again, that he utilizes in this mental health approach. And I guess what I'm getting at with that long preface is, what do you see the potential hopeful impacts this space makes in the mental health and just general physical health field for biology?
So I'd say starting from the cell, once you understand the cell itself, then you can start understanding how that propagates into a larger system, which is called a body, right? But I think, so have you ever heard of Chladni plates? It's like a metal plate that you put some sand on and then rub and make specific shapes.
Yes, I've seen the YouTube videos of this, I think, potentially. Yeah.
So now think of that as one slice times a bajillion, right, is a 3D Chladni plate, right? That is circulating and spinning, right? And instead of sand, it's electromagnetic energy, and it's overlapping with itself. It's making computations. That's called the brain. So when you start understanding what shapes equal what conditions, right? Like what does it look like in terms of electromagnetic energy to be depressed? What does it look like to be bipolar? Which areas of the brain are creating the wrong shapes that when overlapping are creating a feeling, right? I think that that's when you start getting into understanding consciousness, but deterministically, right? Because these are all triangles that are overlapping that can be calculated. But of course, as you can imagine, it's deeply complex. extremely
Oh, yeah.
complicated, right? And that means that the only way to really model the thing is to build the thing, which is kind of funny in that, like, if you want to create a brain inside of a computer and then make decisions off of that model, you basically have to build the brain
Yeah,
because it's a
yeah, yeah,
quantum
yeah.
computer.
Wow. Yeah, so the quantum computing aspect, well, I hear a lot in data and cyber infrastructure and things like that. You hear of quantum computing, and even recently, like I saw that they were finally able, like the longest distance in recorded history, like however many kilometers it was, actually keep two entangled particles entangled at a distance. But you see how incredibly complex it is to keep just two particles at like 40 meters or something, or 40 kilometers. I don't know. Either way, it was like two subatomic particles at 40 kilometers. That was preserving the integrity of a quantum computer in a wet, moist, warm human brain. Granted, it seems to be now, like with photosynthesis, they've been able to show like superposition occur with photons of light. So at least there is some headway. Like maybe the brain is one of the few places where it can be non-absolute zero temperatures, and quantum computing can still occur. But maybe we just don't, like you said, we're not even close to being able to build something as complex as a brain where quantum computing could be safely conducted, maybe.
And I think that what tetronics provides is understanding that you can compute using geometrics, right? So in terms of a specific shape overlap with another specific shape, depending upon how much energy are in that shape and how that energy is arranged, will produce a different shape. And that's the foundation of what quantum computing will be, in my opinion. And that will open up huge amounts of stuff. As in, then you can start understanding that you can do quantum computations using relatively simple things, but it's dependent upon shape. Right now, we're really, really struggling to really do the most basic computations because we don't understand the basic shape of the quanta. And without
Yeah.
that, it's like kind of doing stuff in the dark. You're kind of like doing it a bajillion times and then taking the average. And you kind of end up with the right solution, but it's very, very difficult.
Yeah, yeah, it's interesting too, because we know form and function play a giant role. And even if you wanted to stay in the central dogma, as we were talking about, if you wanted to stay in what people are familiar with, form and function at least is a place to start. It's like, look, shape is absolutely critical, as you already know, in the very specific X-ray crystallography that's been done of these complex three-dimensional proteins. You can see that when a ribosome, it's beautiful, really, the way a ribosome creates and constructs these complex 3D shapes. And you understand that literally if it doesn't fit in the receptor, it doesn't work, if it doesn't very specifically fit. And so at least like there's that structural element to it that can be hounded on. Like, look, shape, if you get shape wrong, it doesn't work. At least here we know that biological systems fail, neurotransmitters don't. Even if you wanted to create an antagonist or an agonist of a certain neurotransmitter, you realize that if it has the wrong enantiomer or whatever the word is, with the wrong analog of that, it won't work. And so it seems to me not too unintuitive or disintuitive to think that, oh, this idea of shape being critical, if you get that wrong, it would be wrong.
Much harder, right? You can still stumble your way to solutions, right? And with a lot of effort and a lot of manpower and a lot of money, you can get there, kind of. But once you understand the basic underlying shape, oh my God, the things we'll be able to do. I mean, I'm a deep believer in humans. I think humans are good. I think we're an incredible, interesting species. What we'll be able to do when our imagination matches one-to-one to reality, which right now it doesn't, in that when you're a physicist imagining or you're a chemist imagining elements coming together, what you're imagining are balls or mathematics, but it's actually a shape. Once we have that one-to-one matching, I think humans will be extremely powerful. I mean, in a potentially scary way, as in when you have that power, when we understood E equals mc squared, we were able to build an atom bomb. When we understand this theory, the things we'll be able to build are not atom bombs, they're planet killers. It's
scary. It's wild. Yeah, it's completely and utterly wild. just wild. The billiard ball idea of particles slamming into each other, and like it still seems like physicists are quick to harp on the fact that the Bohr model of the atom is dead. But I would challenge most people do not think that way. They still don't think of the Bohr model as dead. I mean, physicists probably do. We don't think of it as these SPD and F orbitals with this probability of a particle being here and not there. And you've got, if you get down to the crux of what matter is, nobody knows. And Kelvin's theory does at least cement it in a geometrical shape. It gives something meaning to why do we zoom in on something and it disappears, or it's in two places at once, or it's whatever. That's why I talk about consciousness a lot. It's like this type physicalist where there's a 1-for-1 correlate between this region of the brain and depression. It's not that easy. And the thing is, that is the primary driver, just like the Bohr model kind of seems to still, whether they want to dismiss it or not, it still seems to be driving physics.
So pilot wave theory, I would say, is closer to what Kelvin has stumbled on, and I think it's actually very interesting. There's a Copenhagen interpretation, right, of an electron, essentially. The idea is that the electron, right, so we talk about what an electron looks like. The vast majority of its energy is within its electron cloud. So if you just ignore the particle component and only focus on the wave function of the electron, then you can do a lot of beautiful calculations, right? But you still kind of ignore pieces of it. And that's the problem. The problem is that we've taken this interpretation of ignoring the particle component of an electron and focused only on the cloud, and we've extrapolated it to other things. There are no particles in a way. And that's caused a lot of problems. I mean, it is beautiful in that when you start building up the elements, you can actually see what an SP, D, and F orbital are. They're real physical things that are not just mathematical decisions that we came up with. It's natural. Like, it doesn't matter what we think about the orbitals. Those orbitals are going to be the same on Earth as they are in galaxy X, galaxy Y, galaxy Z. And that, I would say, is kind of opens us up to be able to have a common language that is a physical language, that's a physics language, but there's also a biological language, which I would argue is common across many galaxies. And that lets us start talking about having a common language and being able to talk with things that are living here as well as other places.
Yeah, absolutely. No, and I think when you just said right there about the orbitals being physical things in this tetraionics model, that would be like the SPDNF that we see. We in the valence shell or VESPR, the valence shell electron pair repulsion. They've got these models of shapes, that are tetrahedron being one of them. That when water comes together and you've got this polarity between the hydrogen, you got the oxygen that's pulling the quarter, that's making the model bent. I think that's actually the technical jargon, is the water molecule is bent. And those VESPR shapes, they had the tetrahedron in one of them. These orbitals that they're talking about, and how are those represented in the tetraionics model? Exactly.
Yeah, and again, it's all depressingly simple.
Yeah, I think you may have already explained it. I think I'm having it's hard to grasp for me, but
you'll see it again as you keep hitting it over and over. It gets more and more simple. And that's what is attractive about this. It's not like it's not like French where you end up with like 15 different rules in order to say this. No. So if you look at an element, say you start building from the bottom up. You have one deuterium atom here, another deuterium atom here, and then you build up another level. And then the first level has 2, second level has 8, and then onwards and upwards.
Yeah.
If you look at what an element, just the particle, you build it up, these levels are the quantum levels. There's 8 of them. And then if you turn this and look at it like that, are the SPDF orbitals. Like
okay.
So the
I see.
SPDF orbitals are looking from the top down, and that's as this whole thing is spinning. So as it spins, the electrons are going to have specific radiuses depending upon where they're sitting.
And the electrons are just more quantum coins stacked in different ways that we
The
don't
electrons,
think of
right?
before.
Each deuterium atom is one proton, one neutron, and I wish I had pictures. I'll send them to you as well. I mean,
Sure.
this is all, it all looks
Yeah,
the
and,
same.
and when I talked to Kelvin, he had a bunch of models that he was showing me, and then he emailed me about, so as I got further into consciousness, I got further away from physics, and I'm trying to get back into it.
So I taught myself 3D printing because I needed to make sure that I wasn't crazy. So this is a 3D printed model. This bottom part is a proton. This is an electron, and then this is a neutron. So you see how this spins inside of it?
Yeah.
That's an
Wow.
electron that's spinning. So
And it's made up of all those up-down quarks that are triangles that stick together.
Right. So this is a deuterium. A neutron. So
Yeah.
this chunk here, these three, would be down quark. Then this would be an up quark, and this would be a down quark. So each baryon, protons and neutrons, are made up of 3 quarks, and you can see them. As in, you see how it's made up of 9.
Yeah, yeah.
Neutrons.
Okay, yeah.
So
wow, that's cool.
Right.
Yeah, I mean, it's just cool. Yeah,
It
works, I guess. Yeah.
So these are models that I printed again for myself to be able to know that I wasn't crazy. So see how these are made up of the same thing?
Yes.
And you see how there's electrons in each of them.
Yes, yes.
Neutrons,
and as you stack upward, okay, yeah.
As you stack upward, you get quantum level 1, quantum level 2, quantum level 3. And then you see this is actually would be spinning like this. So this part is the s orbital, then the p orbital. What you're looking at here is, I think, sulfur, phosphorus.
Okay.
Then this would be carbon with 6, for example.
Wow.
And you can see how as you build up, right? It's like not up to me. Like,
yeah, no, I know. Yeah, and then you're just
right.
Stacking deuterium atoms onto each other is what
Exactly.
creating different yeah, and then you're looking
Right.
at it, like you said, you're looking at it top down, then you're looking at it plan view or whatever you want to call that.
Right. And this would be, for example, this would be oxygen. You have 1, 2,
3, 4, 8.
Right.
Yeah. Okay. That's
and
cool. That's neat.
Different shapes, right?
Yeah, yeah, yeah.
And the different shapes will influence how it pairs up with other things, right?
Yeah, yeah. Those tactile models, the 3D printing thing, that was a great idea. And
I mean, I like that.
Well, yeah, no, yeah. At that point, yeah, I understand that, but that's cool that you did that because.
And then, like, for example, like one of the other ones is like when you can start doing chemistry with these, but it's quantum chemistry. You actually start, right? You think about the as these electrons are spinning, right? When you pass, as these electrons are spinning, they're creating huge clouds around them, right?
Mm-hmm.
And the clouds will have different shapes according to the underlying shape of the element itself. Mm-hmm. And another thing that to me was interesting, so you see how this is colored? They're colored according to the quantum levels, because when you spin, for example, you spin this bottom electron, is it going to cause this electron to spin?
Yeah.
Is there going to be energy that is transferred from one quantum level to a second quantum level?
Yeah.
And that's called emission spectra. So it will emit a specific energy that is the difference between this quantum level and this quantum level. That difference is the frequency that is being emitted of photons.
Wow, so that's really and that emission spectrum idea and everything is an understood facet of chemical interaction and stuff, but it's not envisioned with a model that's tactile and that's physical and has a shape like that.
That actually has
instead of this quantum foam of improbable events that may or may not occur, it's eventually you've got to ground it in. If you want to call yourself physicalist and materialist, you've got to ground it in something physical and material. That would help, and that's why it's so hard to come up with stuff from the physical. Whenever you're looking at strings, or you're looking at something oscillating and stuff, there's nothing grounded. And like you
Right.
said, this
It's all
model does
mathematics.
that.
And I think that science is going through a real problem right now where there's no grounding reality, and people have gone off the deep end. There's a lot of people that have gone off the deep end, even very well established people, et cetera, when you don't have an underlying physicality. And I think this phrase, Kelvin came up with it, which is tetraionics is the grammar of physics. So you can write an entire novel that has no sensical to it, but it is a novel. But what tetraionics lets you do is understand that there is an underlying grammar to this structure, and it lets you start thinking about these things in a physical way, right? That you can imagine as a person, but as you can imagine as this thing is having all these electrons spinning at different frequencies, and it's coming into contact with different things, and it's spinning like this, it's creating different clouds. It's very difficult to model, if not basically impossible, without a quantum computer, which is also stored in our brain, right?
Yeah, which is conveniently there. And then that's so neat to see how you came to tetraionics through the way that you did.
I mean, I had to build it up. I did not know if this theory was useful or was able to provide testable theories, right? And then as I built up, I was able to start seeing that the things that were getting built were the things that were getting measured. And at that point, I needed to do that, to make sure that it worked.
Otherwise, to ensure the rigor's there. I mean, to ensure the scientific backbone of the whole thing, which test and observation, and then having that confirmation of that is incredible. Really, it's really fascinating. And you see this has come merging into your space of biology, but I mean, you've broadened your scope of knowledge, or scope of which is cool. Right now, what are you working on right now? With this, I'm just curious. What are your hopes and dreams for, or what your space is in? I don't mean to pry into it. I mean, I know it's your personal life. It's fascinating, and I was curious kind of what your ambitions and aspirations are for what you're working on now.
Yeah, so I mean, I want to get this knowledge into the general populace, because it's like, if not me, then who?
Yep.
So trying to get it out to people. You're the first person that I've really talked to about this kind of thing in a more public setting. I'm a little afraid, as in, like, when this stuff gets out, I may face more backlash. I don't know. We'll see. So there's the tetraionic side of things, that what I'm particularly working on is trying to come up with some type of battery that is based off of the understanding that as you spin those electrons up, the energy is stored as matter, kind of. As in, it doesn't get heavier, but it does get heavier. Does that make sense?
I see how it makes sense. Yeah, in a way. Yeah, I see what you're saying. So that's kind of where you're trying to go, that avenue with it, is to trying to solidify this theory in some way or another by that route.
Right.
Yeah.
To be able to create something that is unarguable. Like, it works, so how do you explain it? Well, I can explain it, and it's pretty simple using this theory. That's the tetraionic side of things. Now, in terms of the biological side of things, I stumbled on a loci at MIT that I think defines what viruses are your own versus everything else. Once you understand that the innate immune system has a link to the adaptive immune system, this is when we're talking about T cells, VDJ recombination. I don't know if you're familiar with that kind of thing, but what it lets you do is define very clearly what is cancerous and what is not. So it should open up the ability to design, I hate saying cancer cure, but it is understanding how our endogenous cancer system actually works, and then playing around with it. So those would be my two biggest kind of focuses. But
cool.
in reality, as you can imagine, this opens up so many things that's really what I'm interested in, and people taking it, moving forward, creating crazy stuff. I want to see a Star Trek universe, and I want to see it within my lifetime. And this
Yeah.
is the way to do it.
No, I like it. I love it. Awesome. Well, thank you a lot for talking with me today. I'd love to talk with you more in the future. I'd love to talk with you and Kelvin sometime. That'd be excellent. It'd be really cool. I've kind of gotten off on to the consciousness, but I've tried to stay in consciousness studies, interviewing people, reading these books, talking with as many people as I can on the consciousness and physics and biology and chemistry. I've tried to keep, I've talked to like about 30 people thus far since I've started doing this podcast, at least, and read probably 4 times that amount of books, but that's basically what I've devoted my time to for like almost completely. But
good. You mentioned Cartesian coordinates. I mean,
yeah.
Rene Descartes said that the pineal gland is the seat of the soul. I think he's right. And the way that
he's right too.
Typically works is by using, so there are 2 areas in the human that have crystals. One of them is the inner ear, otoliths. The other one is the pineal gland. There are these crystals that are calcite crystals that are, oh my God, I forget the word, but when they're exposed to an electrical field, when they're exposed to pressure, they create an electrical field. So what I think is happening there is basically an inner ear, but for electromagnetic frequencies. And that's sitting in the middle of your brain and causing all these brain waves to split. It's a birefringence is the word. So if you take a calcite crystal and you look at like a letter, it will split that light into 2. So what this area is maybe likely doing is actually increasing our ability to be a quantum computer, but a quantum computer that can actually listen to things outside of itself. And that's where you get into really crazy stuff in terms of when we're able to listen to electromagnetic fields that are outside of ourselves. It means that we can start intaking information that may not just be within ourselves. And then you get into dreams, and you get into the tryptophan pathway, and then you get into how DMT is produced in the brain. And I mean, it is wild, but I think it's all a measurable system. But you need to have a very foundational grounding in order to be able to ask these questions. So
no, that's neat. Yeah, the pineal gland, it's such a fascinating thing, and Descartes was a really fascinating. Descartes really responsible for a massive philosophical worldview shift, and this from idealistic looking at the world as consciousness first, and material to material first, and consciousness second, and this physicalist universe emerging that we view the world in nowadays. We see as one of my favorite philosophers, who's a computer scientist, and he's got a PhD in computer science, Bernardo Kastrup, who's specialized in what he wrote like 150-page thesis on his that he defended. There's a YouTube video of him actually defending this thesis for his PhD, but it's called analytic idealism, and it's a way of trying to formulate a worldview that keeps the physicalist. But his argument is that Descartes strictly separated and placed quantities before qualities. That before we saw qualities of mind, qualia of experience, the feeling and subjective element of being set first. We had in mythology, there's a lot of animated plants, there's a lot of animated animals talking and stuff, because it was natural to see like this idealistic worldview where like the sense and subjectivity came first, and then we gave rise to like objects of perception. And Descartes, with his mind and body split, the pineal gland was the way in which everything was grounded in quantities that gave way to qualities second. Qualities came from the rearrangement of quantities of matter. And Descartes is paved the way to this worldview we have now. So it's highly logical to assume that his pineal gland and its psychedelics and everything are wild, and psychedelics and the just subjective experiences I've had with those and stuff. Something goes weird with the pineal gland, that's for sure. I just tell you, I mean, there's something active about it in psychedelic states and stuff like that.
Like,
so anyway,
why do we have to sleep? Why, like, why,
Yeah, why do we
why,
have to sleep?
if we don't sleep, we freaking die. Like, it's more important than eating. So why,
Yeah.
right? These
Yeah,
are
why?
questions that we need to explore. Otherwise, again, I think we're screwed.
Yeah, I agree. Well, Diego, thank you very much for coming on and joining me. Spent a little over an hour, but I appreciate it, and I hope we can further and talk more in the future, if you don't mind. And
Absolutely.
we'll kind of drive further on this, and I'll hopefully I can get in touch with Kelvin again too. Have you talked with Kelvin much? Just real quick.
No. So that's also one of the things that's interesting. Like, I stumbled on his theory and I needed to make sure that it was rational before I talked to him. Now I've talked to him on Facebook a couple of times, but no, I intentionally kept some kind of boundary between us because I didn't want to be influenced. I wanted it to be an independent, semi-independent conclusion. And at this point, I now have reached the point where it's like, guys, let's get together and start building some stuff.
Yeah. Because when I talked with him, it was like 3 hours. It felt like maybe 2.5. But I was just coming in on the base level of having. But I stumbled into this by really having this just intuitive desire to figure out what about will, agency, and motion, and cellular biology, and especially microtubules and the cytoskeleton. What element those have to, because those are quantum computers. The cytoskeleton and the tubulin fibers are quantum computers that resonate and reverberate at certain elements, and if you administer anesthesia to even the most elementary of prokaryotic organisms, you see, and they go into a state of anestesis, not a state of sleep, but a state of non-responsive to their environment, an actual anesthetized state. And if you can anesthetize a single cellular prokaryote, that means that you're taking away consciousness. So that means consciousness is there. Well, what about that process of anesthetization is happening at the cellular level? Well, it's the interruption of the microtubulin fibers to actually be in a dipole state. So that constant dipole switching in the microtubulin fibers, they got 8 tube, and that switching on and off and changing seizes up and freezes in anesthetized prokaryotes. Same thing as it does in the human brain. And so I was like, well, there's got to be something to this.
Right. And then you get at like, so I just left an agrochemical company because I needed to understand plant life better. And then the whole story gets really freaky when you start talking about the tryptophan pathway. I mean, tryptophan pathway involves serotonin, ends with dimethyltryptamine at the end, and makes you trip. Now, what's really bizarre is that plants also have a tryptophan pathway. Why? Why do plants have a tryptophan pathway? Why?
That is why.
So these are things that, again, we need to figure out.
Yeah,
and it's exciting. I mean, one thing that people have kind of gotten in their heads is that when there's great discoveries, it solves everything. That's a lie. Whenever there's a great discovery, it lays a foundation to branch out like crazy.
Yeah, no, yeah, it definitely opens a lot of doors, but doesn't tell you what key's going to unlock them. But your key's out there now. You have some sort of semblance of a combination that's going to unlock them, and that's even more exciting than unlocking them because because you don't know what exactly door you're going to open, where it's going, but it's going somewhere positive as long as you're on the right conscious plane and you're not malevolently going and searching for these answers to like extract vengeance on your foes, which we're not. You, Kelvin, and people like that none of us are doing that or trying to figure cool things out and in such really vast ways. And you and Kelvin and your partnership with his theory and everything, I think it would be cool one day to try to because Kelvin was super nice to me afterwards and everything, and was telling me, anytime I wanted any more questions arose, to just freely reach out to him. And he's in Australia somewhere, but he makes the time, and so I think it would be cool to maybe chat with him and see if he would like to talk with all three of us one time. That'd be cool.
Yeah, definitely. Let me know.
Yeah, I will. I will certainly do that. But that's great. Thank you so much again. I'll go ahead and let you go, but I'll be ruminating on this for a while, and I'll
play my game. Try it.
Yes, I will definitely do that. I'm
Let me know
excited.
if it works, because I've been building it for a while and I've been crashing computers the whole time.
Okay, well, I will do that, and I'll keep in touch with you, and we'll see what we can do in the future.
Cool. Sounds good.
All right. Thanks, Diego. I'll talk to you soon.
All right. See ya.
Bye.